Good evening, everyone from the US, and good morning to those of you that are in Australia. My name is Suzanne Barnett, and I'm hosting your webinar for you today. Now, before I get right into the webinar, can I just check that everybody can hear me first by clicking on the hand button on your computer to indicate that you can hear me fine. Thank you, Becky, thank you, Natalie, thank you, Lisa. But a few people are coming in as we've started, but it looks like everyone can hear me fine. Sometimes I don't see a show of hands, and then I have to perhaps assume that you can hear me, but if you could just type in to the question box just yes, that would be great. That would also help. Okay. And sometimes it depends on your on the particular handheld or computer that you're using. Perfect. Thank you so much for responding. So it looks like everyone that's here can hear us perfectly fine. So like I say, my name is Suzanne Barnett. I'm hosting your webinar for you today. And I would really request that you switch off other applications that you've got on, because it does improve the quality of the broadcast. And we are aiming to finish today by 1 p.m. We have run over in the past, but we are aiming to finish around about 1 p.m. We have allocated some time at the end for some questions, so do fire those questions out as you think of them, and then I will put them to the presenters at the end of this webinar. So our topic today is methylation and methionine. Our speakers again today are Sterling Hill, Carolyn Ladowski, and Cynthia Smith. And our first speaker today is the lovely Cynthia Smith. So if you would like to start with your part of the presentation, that would be great.
Thank you. So today we're going to focus on methylation and methionine. And if you, most people are pretty familiar with those pathways, and they interconnect through MTR, and then the methionine pathway also has some outputs like SAMe and some other things. But today we're going to just focus on those two pathways. The folate methionine cycle eventually will connect back to the biopterin cycle through the transsulfuration pathway output, and also the urea cycle. So let's go to the next slide. This is the Seeking Health Pathway Planner. If you'd like to get copies of this, Ben Lynch has them on his website. But today we're going to focus on the pathway on the right, top right, and then the pathway on the top left. And we're going to start with the folate pathway. So when we're talking about the two pathways, again, you see the folate pathway on the left and the methionine on the right, interconnected with MTR. Sterling's going to be talking to us about the various SNPs here, but I just want to point out that DHFR is the first enzyme that deals with folate or folic acid. So if you eat something with folic acid, you'll be using 2 steps of DHFR. If you're eating something with folate, you're only going to be going through 1 step of DHFR. And then there's a 3-step MTHFD1 pathway that leads you into MTHFR. There's a side branch from MTHFD1 through SHMT1 that creates folinic acid. Many people have SNPs and MTHFS, and in that case, sometimes folinic acid is contraindicated. Now, TYMS, and I don't know if we're going to talk about this today, but TYMS is a feedback, and there's a branch off of it that's not shown here for DNA synthesis. And so TYMS SNPs are important, as are getting through the steps through MTHFD1, so you have the building blocks to make your DNA. Okay, let's go to the next slide.
Cynthia, sorry, I'm sorry to interrupt you. I just need to check that everybody can hear Cynthia fine. Just give me a show of hands. And Donna, could you just type to me that you can hear Cynthia? And I've also got a comment that someone can't see the slides, so I just want to make sure that everybody else can. Okay, so first of all, people can hear me. That's good. Anna, thank you. Thank you. All right, so I'm going to put your hands down. Now just tell me, can you see the slides, please? Just with a show of hands and a yes. Yes for the slides. Okay, so Donna, you may not be able to see the slides because you're not looking at the webinar system itself. If you click on the daisy at the bottom of your screen, you'll see that the slides will pop up for you because everybody else can see them. So thank you for that, Cynthia. Sorry to interrupt, and you can carry on now.
Well, I think Sterling's up next to talk about the various SNPs.
Okay.
Hello, everyone. Okay, I'm going to talk about the folate receptor genes first. Folate receptor 1 is known as folate receptor alpha, and it is expressed in adult tissue. Folate receptor 2 is known as folate receptor beta, and it is expressed in fetal tissue. Folate receptor 3, known as folate receptor gamma, is expressed in embryos and also adult tissues. Now, with the folate receptor genes comes cerebral folate deficiency. We see that in a lot of children with autism. And one of the things that we've learned about the folate receptor genes, the folate in the brain, the folate receptors in the brain, is that folic acid will actually prevent folate from crossing the blood-brain barrier in these children. So folic acid really, really must be avoided. It's important to know. Now, DHFR, dihydrofolate reductase, this is an enzyme that reduces dihydrofolic acid to tetrahydrofolic acid using NADPH as an electron donor. Again, now I want to stop here and talk about G6PD. G6PD is not functioning. You see someone with G6PD deficiency. Note that the dihydrofolate receptor will not be working because that's all about NADPH and the pentose phosphate pathway. That's not functioning. DHFR will not, because it's also part of the pentose phosphate pathway as well as this pathway. But it converts tetrahydrofolate, every kind of tetrahydrofolate there is, so it's really important. Now, MTHFR, again, that stands for methylene tetrahydrofolate reductase. MTHFR is really important, and we always hear every practitioner, they go to people, they see somebody with MTHFR in their variant report and they throw them folate. That is wrong. First of all, MTHFR is the last SNP that needs to be addressed in methylation. So you look at everything else first. You got to make sure other things are in place. Again, folate is not the only thing that's needed for MTHFR to function properly. The other one is NADPH. Again, we go back to G6PD. If G6PD is not functioning and you have a G6PD deficient person, then methylene tetrahydrofolate reductase will not be working properly. So it requires not only methylene tetrahydrofolate, but it also requires NADPH. Another thing that I want to get with on this one is giving high-dose methylene tetrahydrofolate to somebody with homozygous COMT, and COMT we'll get to later, I think next week or week after, the COMT gene, when they're homozygous for V158M and H62H, you could sometimes cause quite a bit of anxiety in those people. Now, when someone has a defect at one of the MTHFR enzymes, he or she can have an array of problems related to folate deficiency, such as neural tube defects, spina bifida, elevated homocysteine, poor methylation issue, miscarriage, cancers, and autoimmune diseases. Now, just because you have MTHFR does not mean that there's a problem there. And that, again, this is what I want to get through to the practitioners. You can't treat the SNP, you've got to treat the person. We have about 12, 13 SNPs on the variant report that have peer-reviewed research attached and that show relevance to having a problem with MTHFR. 98% of the population has an MTHFR defect. 98% of the population is not folate deficient. So keep that in mind, please. Now, we have seen also with MTHFR, of all the SNPs that we do have, I've had about a dozen people that have been homozygous for more than 10 mutations. About a dozen people out of thousands. And if you ever come across this, check them for tetrahydrobiopterin deficiency and low dopamine, because that's what we saw they all had in common. Very low dopamine. Some had dystonia, some had Parkinson's, and they were all tetrahydrobiopterin deficient. So that's a good thing to know. Again, when folate is deficient in someone, you can have some autoimmune issues. Folate controls natural killer cells. So when natural killer cells become low, you can have autoimmune diseases as well. When it comes to miscarriage and MTHFR, that one's really important too. I learned this from my hematologist when they found out through looking at my blood under a microscope that I was folate deficient, the way my red blood cells were shaped when I was in the hospital with pulmonary embolisms. He told me something interesting about it in miscarriage. He said that women that are folate deficient, their red blood cells will become oblong, misshapen, get hooks on them, and capillaries feed the uterus. So with the capillaries feeding the uterus and the misshapen red blood cells can't get through to feed the uterus, you're going to have miscarriage. That was pretty interesting to learn that. And something so simple like a change in diet could help these people, or a little bit of folate. And we've had many people call me up. I've had many people call me up and tell me, thank you so much, because of you, I can have a baby now, getting this information out here. And it feels really good when I get those phone calls. It's so important to know that folate is really important, and folic acid is different from folate. MTHFR, again, the immune system, it's a big one. Keep an eye on that. And if you go and you give somebody folate before you address other things, you can really cause a jam up in there. I, for one, myself, I took some high-dose methylfolate, methyl B12 mix, and I felt really good on the first one, actually a little too good. I looked like I was high as a kite. I went from chronic fatigue to bouncing off the walls. And when I took the second dose, my bones were hurting so badly that I ended up in a hospital for 6 days on morphine. My white blood cell count went from low to majorly high, and I turned on things too fast. I was jammed in other places. I also had homozygous compt, which is not a good idea for people to take high-dose methyl anything with the word methyl in it, since their enzyme regulation is really poor for breaking down dopamine, epi, and norepi. Again, MTHFR, really important one, but always remember it's one of the last SNPs you address. Now, MTHFS, that one right there, MTHFS, methylene tetrahydrofolate synthase, that one requires magnesium support. And as Cynthia talked about earlier, it's really important to know that these people sometimes cannot, probably most of the time, cannot handle folinic acid. I believe Carolyn has told me in the past too how she went and tested, and many of those individuals are folinic acid, high in folinic acid. They're not breaking it down. MTHFS is also related to kidney failure. One of the reasons it is because it requires magnesium. These people need a lot of magnesium support. Oh, you always want to check their magnesium. Now, SHMT, I got this information from Dr. Yasko. She's actually a very brilliant lady. She's the one that pioneered, brought out to the public the methylation pathway. And I would just like to read just a little bit of what she says about SHMT. This gene product helps to shift emphasis of methylation cycle toward the building blocks needed for new DNA synthesis, away from the processing of homocysteine to methionine. While DNA building blocks are important, mutations which affect the ability to regulate this gene product and interfere with the delicate balance of methylation cycle may cause accumulations in homocysteine as well as imbalances in other intermediates in the body. Serine hydroxymethyltransferase is a pyridoxal phosphate containing enzyme. It catalyzes the reversible conversion of serine and tetrahydrofolate to glycine and 5,10-methylene tetrahydrofolate. Folate-dependent 1-carbon metabolism is critical for the synthesis of numerous cellular constituents required for cell growth, and SHMT is a central process to this. Okay, I'm done, guys. I think it's Carolyn's turn, right?
No, Cynthia's going to go
I think
through.
this one's my slide. So when folate. So when we're talking about folate, we not only have to talk about the receptors and the series of enzymes, DHFR, MTHFD1, MTHFR, we also have to talk about the transporter proteins and some of the membrane proteins that regulate intracellular folate. There was a study done about women who were trying to get pregnant. They did use mice in the study, but for those women who had accumulation of defects in not only the folate receptors but also some of the folate conversion enzymes, they had impaired nucleotide biosynthesis and so impaired repair of DNA. What happened in those mice is that the embryos died, and sometimes they died. They lasted up until it was time for the neural tube to close, but they didn't flourish. If you gave those same mice 5-methyl tetrahydrofolate, the pups were normal and there were no abnormalities. So I really think that nutrients are right for some people and not others, and it's not one size fits all. You have to look at the receptors, the transporter proteins, and as Sterling mentioned, the nutrient cofactors for these. In people who are taking folic acid, it oftentimes preferentially binds to the folate receptor and will actually block the activity of absorption of folate. So folic acid is really not good for people that have SNPs in those receptors. This next slide is a simplified drawing of the folate pathway, and we're looking at folate receptor 3, folate receptor 1, and you can see how folic acid will preferentially bind to the folate receptor 1, and we do not want that. Folate is really important for rapidly growing cells. So again, that's pregnancy, a fetus growing. Also, during times of repair, if somebody's had surgery or they've had some sort of an injury where the body's really working hard to make new cells and repopulate their DNA in the cells, they really have to be conscientious of their folate intake and try to make sure that they're keeping this pathway open. The primary role for the folates as far as DNA synthesis was established in the 1970s, and we all know that 5-methyl tetrahydrofolate is the most available form. But again, as Sterling mentioned, we have to be careful when we combine that with methyl B12 if certain genetics are present. Now this next slide talks about folate receptor and dairy. Folate receptor antibodies are increased with dairy. So what that means is if you're drinking a lot of cow dairy, especially in the United States where we've got all kinds of junk in the milk, you're actually downregulating your folate receptor. You can even develop antibodies against the folate receptor, and therefore you're kind of destroying your folate receptor. So people, especially with autoimmune conditions, if they're on that autoimmune spectrum and they have, let's say Hashimoto's or they've got rheumatoid arthritis, they're particularly vulnerable to this because their immune system is already upregulating from other things. Now the next slide is specific to the folate receptor 3, and as you can see here, the risk allele is G. And this is a risk for lack of closure of the neural tube during embryonic development, and that's about the time when the ball of cells, when the sperm and egg come together, the cells start dividing and you have this ball. I think at about day 14 that ball turns inside out and starts to form the cleft, the spine, and on down. And so this is where a person can get into trouble if they've got this folate receptor SNP and if they're low in folate as well. Now DHFR is a key enzyme in folate metabolism, and as Sterling mentioned, it converts dihydrofolate to tetrahydrofolate. So that means 2 to 4, and it's a methyl group shuttle for other components. DHFR is a one-step process for folate and a two-step process for folic acid. But again, we don't want to encourage anybody to take folic acid, and you have to be careful with folic acid because, at least in the U.S., they use it in all kinds of processed foods. It's in bread, it's in cereal. It was the way that our government tried to stop the spinal bifida and some of the other things like cleft palate back in the day. Dihydrofolate reductase is a methyl group shuttle required for synthesis of purines and certain amino acids. And so again, this has multiple functions. One of the byproducts of the DHFR enzyme is a feed into the biopterin recycling, and we'll talk about that another day. This enzyme also catalyzes the reaction for de novo glycine and purine synthesis. So again, it's part of the whole DNA building blocks. This is just another slide talking about DHFR. Again, the biosynthesis of nucleotides for incorporation into DNA and RNA. Pathway 2 shows you the remethylation of homocysteine to form methionine with that intersecting MTR enzyme. And the methylation substrates, including RNA, DNA, phospholipids, and Carolyn will talk more about the phospholipids when she talks about the methionine cycle, and proteins. Now the next slide is another SNP indeed that we see in DHFR with the risk allele of A at that particular RS location. In this study, the authors hypothesized that polymorphisms in this gene contribute to the prognosis of non-small cell lung cancer. The authors screened 57 potentially functional polymorphisms from 11 candidate genes involved in this one-carbon metabolism and genotyped the variant alleles in 568 patients using a particular method called Illumina Golden Gate platform. But anyway, the variant alleles that were significantly associated with favorable survival were MTR, RS3768160, which again is that intersecting enzyme. There was also a link between these SNPs and skin cancer. This is an article that talked about the variable activity of dihydrofolate reductase in the human liver and its implications for high folic acid intake. And again, it's talking about the competition and how you can downregulate that DHFR enzyme with folic acid, and we don't want to do that. Now the next slide is again about DHFR, and there's a concern that the presence of unmetabolized folic acid is detrimental. And this particular study suggests that it decreases natural killer cells. And as you know, natural killer cells are very important for the immune system, and it's the natural killer cells that are often suppressed when cancer cells start to take a foothold. So a high intake of folic acid can exacerbate some of these pre-existing cancers or the progression of cancerous lesions in areas specifically like the colon or the prostate. So it's not surprising that we haven't seen the results from the clinical trials that we expected, because many of them were using folic acid and not 5-methyl tetrahydrofolate. So when you look at these studies, sometimes you have to look carefully to see what they're basing their conclusions on. Now, what affects DHFR? So we talked about folic acid blocking and slowing it down. Too much of it can saturate the receptors. Methotrexate is a drug that's used for autoimmune disease, and it actually targets this DHFR enzyme and slows it down. So what they're doing is they're slowing down methylation. Green tea can also inhibit DHFR, as well as grapeseed extracts, polyphenols, which we know are good, but we have to balance this out, right? You need to know what your genetics are before you start taking some of these supplements. Its cofactor is NAD, because there's a lot of interaction between NAD, which is from vitamin B3, NADP, and NADH, and we'll learn more about that later when we talk about the pentose phosphate pathway. MTHFD1, this again is a 3-step process. So it converts tetrahydrofolate to 10-formyl tetrahydrofolate, and then out of there are made purines for DNA synthesis. The next step of MTHFD1 converts the 10-formyl to the 5,10-methylene, and out of that there's a feed forward into SHMT, and then folinic acid, and then the feedback through MTHFS. And then the next step is the 5,10-methylene to 5,10-methylene tetrahydrofolate, and then that's what goes into MTHFR. So this one has 3 steps, and each step is important. And when we're looking at SNPs and MTHFD1, we're looking at SNPs in all 3 of those areas. Here's another slide of the folate metabolism with MTHFD1. We can just skip this one, but just understand that two of the important outputs are the purines and pyrimidines synthesis. Purines and pyrimidines needed to synthesize and repair DNA, and also, we haven't really talked about this, to methylate our DNA. Methylating our DNA will cause its expression to change. It's like toggling software, or it's like toggling hardware on and off with software. Again, this is another drawing. This is really better for reference only. It might be too complicated to talk about right now with the time that we have, but this is a good drawing for reference, and you can see how, for example, the different, you can see the enzymes like MTHFD1 and see how the NADPH and FAD and ATP are all part of this. So again, we need good Krebs cycle function. We need our vitamins B1, B2, B3. We need a good pentose phosphate pathway, because these are all interacting with this folate pathway. Now, the risk alleles for MTHFD1 and MTHFL, in both cases for these RS numbers are A. Going along with DHFR and MTHFS, we can have issues with elevated plasma homocysteine. We can have issues with folate deficiency. We can have issues with the folate cycle in general, and then of course neural tube defects. There's a lot of research that links this to Alzheimer's, heart disease, and so this is a really important pathway. Now, the link to heart disease, they usually link up to high homocysteine levels, and Sometimes people have high homocysteine levels because of lack of folate, or sometimes lack of B12, or both. Again, this is another slide. The next slide talks about the links to MTHFD1 SNPs, which are different types of cancers, and also immune deficiency, autoimmune deficiency, or autoimmune issues, spontaneous abortions. Now, I did research a long time ago, probably like 2 or 3 years ago, about MTHFD1, and there's quite a bit of research associating MTHFD1 with miscarriages. So that's something to look at as well. And then, of course, the neural tube defects, Parkinson's disease, but we'll talk more about Parkinson's disease too, because there's a link there with dopamine, which is one of the pathways that come off of the folate cycle is this biopterin recycling. And biopterin is a cofactor for making dopamine and serotonin and some other things. And so that's how the link is connected here with MTHFD1. SHMT again is an enzyme that is it picks up 5-methylene tetrahydrofolate between step 2 and 3 of MTHFD1. There's 2 forms. There's one which is in the cytosol, and then there's 2 which is in the mitochondria. And our mitochondria are the organelles that generate ATP. SHMT can convert glycine to serine and back and forth. So sometimes when folks are low in glycine, they could look at their SHMT SNP to see if maybe there's a problem there, and then you can supplement with a glycine amino acid. Remember that glycine is also important for phase 2 liver detoxification. So there's testing you can do to check your levels of amino acids, and so if you've got this SHMT, look at your glycine levels. These reactions also play an important role in neurotransmitter synthesis and metabolism, because glycine can act as an inhibitory neurotransmitter in the central nervous system. And I know about 5 years ago I was at a IFM symposium, and they were handing out little paper envelopes of glycine as sugar substitutes. So you could put it in your coffee or tea because it just has a sweet taste. But they were also encouraging people to take glycine as a way to calm down their, excitatory neurotransmitters, particularly their NMDA receptors from excitation, so that they could get to sleep. Glycine is also an important constituent of collagen, and also glutathione. Glutathione is a tripeptide, and glycine is one of the amino acids. And most people will take N-acetylcysteine to build up their glutathione, because the cysteine is usually the lower of the 3. But if somebody's got an SHMT SNP, it may be that it's the glycine that's the weak link in that glutathione making chain, so to speak. So don't short shrift SHMT. It's very important. MTHFS is the only enzyme, known enzyme to metabolize folinic acid, and this becomes very active during pregnancy. You'll see a lot of the prenatal vitamins include folinic acid because of the increased cell growth and increased need for making purines and pyrimidines and things like that. But the cofactor here is ATP and magnesium. You'll see magnesium has a lot of cofactors in the folate pathway. ATP is something that's generated by the Krebs cycle and the subsequent electron transport chain. Now, in order for the Krebs cycle and electron transport chain to work well to make ATP, you have to have adequate amounts of vitamins B1, B2, B3. You have to have, you don't want a lot of heavy metals, because those will kind of muck up the Krebs cycle and slow it down. And then that could impact this MTHFS. But suffice it to say that this MTHFS enzyme becomes very active in pregnancy. And it's often associated with lower levels of plasma and tissue concentrations of folate as well. Folinic acid has been observed to promote higher levels of glutathione and dopamine in autistic children. And since it doesn't require the actions of DHFR for its conversion, it may be a good adjunct for those folks. But you just have to really look at MTHFS to make sure that you're not flooding the body with folinic acid. I have a note here that Pure Innovation Activated B is a good place to start for those in Australia who can't use methyl tetrahydrofolate. So that's a folinic acid supplement. This is a slide that shows how MTHFS binds 10-formyl tetrahydrofolate tightly and delivers it to the purine synthesis pathway. In other words, delivering it to the DNA making pathway. Here it's referred to as the purinosome, which is a hypothetical assembly of folate enzymes that support production of purines, and specifically thiamine. So this is another slide that's kind of for reference only. As we've mentioned, MTHFS is the only enzyme that metabolizes folinic acid. Therefore, a deficiency in MTHFS can lead to a buildup of folinic acid in the cells. And folinic acid can regulate folate metabolism by inhibiting other enzymes like SHMT. So it's like a feedback. It's a negative feedback loop. So a deficiency may lead to folate levels rising, feedback and inhibit SHMT, and serve to lower the 5,10-methylene tetrahydrofolate, which is then fed into MTHFR, so you'll end up with a lower methylfolate. So you have to really balance this out. MTHF deficiency has been linked to diabetic neuropathy, neural tube defects, as many of these folate enzymes are, seizures, autism, and a reduced risk of reduced survival in lung cancer, and also the cardiovascular disease link. Sterling talked a lot about MTHFR. Everybody knows about MTHFR. MTHFR takes 5-methylene tetrahydrofolate and converts it to 5-methyl tetrahydrofolate, which then passes through MTR via what we call the long path, or what Dr. Yazgo calls the long pathway, and ultimately through methyl swap converts to methionine, and then Carolyn will talk about that. And then the methionine converts to SAMe, which is SAMe is like the big prize at the end of all of this, because SAMe is the master methyl donor and contributes the methyl groups for DNA synthesis. It works with the GAMT enzyme for energy and muscle production, and then it converts phosphatidylcholine or ethanolamine is used by, let me just say it this way, all of the enzymes that end with MT require SAMe. They require a methyl group in order to do their job, and there's probably about 200 of them. Mutations, again, in MTHFR, MTR, and some of these other ones that are intimately associated with that methionine pathway can cause homocysteine, higher homocysteine levels. Here's another nice drawing that shows, let's see, let's go to the next slide. This is a drawing that shows what can happen when you start to lose some of your ability to make these conversions, and how the blocks can start causing buildups of, for example, homocysteine. Now this slide is self-explanatory. These are the different risk alleles associated with the different RS numbers, which I like to analogize to rungs on the DNA ladder. And again, you can see all the different risks associated with it. Some of them are direct and some of them are more indirect because of the interplay with these other folate enzymes. But the big push, like I said, for including folic acid in processed foods was to address spinal bifida and cleft palate. again, the next slide, same thing. There's different studies associated with all of these. Many of these are in the SNP Compendium 1, where we've got the actual research associated with each one of these alleles. MTHFR C677T is one that everybody knows. Homozygous there will impact the folate cycle pretty heavily and reduce the ability to convert to 5-methyl tetrahydrofolate to about 40–50%, maybe even lower, depending on the MTHFD1 and DHFR status, the SNPs there. And then here's the next slide is just a list of references for you to look up. Clinical pearls. There's a 24-hour test to screen for methyl cycle abnormalities. If you detect higher ammonia, there's a problem with the CBS, BHMT, or NOS pathways. Now, when CBS is upregulating, you're by definition probably going to have issues in that NOS pathway because there's such a drain on NOS and things like arginine to get rid of the ammonia that's generated by this upregulation of the CBS pathway. If it's sulfate up, it could be a problem with CBS or BHMT as well. And if sulfate's up with low blood homocysteine, there could be a problem in CBS. I just want to point out that homocysteine, even though it's recycled in the methionine pathway, it's only broken down through the CBS pathway. Some people with low homocysteine have issues just like people with high homocysteines. They're different issues, they're for different reasons, but low homocysteine is not a good thing either. Here are some things that inhibit the folate SNPs, or sorry, that don't inhibit. These are some things that help, that are cofactors, and then a couple things that inhibit. So magnesium, NADP, which are derivatives from vitamin B3, and vitamin B2 is needed for MTHFR. Now, when somebody takes SAMe, because that's often something that people will take when they have neurotransmitter issues or they're a little depressed, you have to be careful with SAMe. Sterling mentioned that if you have COMT, you don't want to take anything that's heavily methyled, right? SAMe is one of those things. It's methyl. It's the biggest contributor of methyl groups. And so if somebody begins taking SAMe and they have COMT, that's a no-no. If somebody begins taking SAMe and they don't have COMT SNPs, they could inadvertently start to slow down that pathway again through negative feedback. The body says, look at all these methyl groups. I don't have to make them. And so you start kind of backing up your folate pathway and your methionine pathway. So SAMe is an important adjunct for some people, but you have to be very careful with it. People who have an intolerance for SAMe, besides feeling increased anxiety, might also start having gut issues like cramping in their upper GI, almost like serotonin poisoning kind of a feeling. So be careful with that. And then inhibitors, of course, are the green tea that we talked about. Even though it's supposed to be, green tea is healthy, ate the component in green tea. And then, of course, folic acid. And now Sterling's going to start us off the methionine pathway, going through the methionine cycle key SNPs.
Okay, MTR, methylene tetrahydrofolate homocysteine S-methyltransferase. That one right there, I'm going to take a little bit from Yasko again too, because she's one of the people that brought this to the forefront for us. The remethylation of homocysteine to form methionine is catalyzed by cytoplasmic enzyme 5-methylene tetrahydrofolate homocysteine S-methyltransferase, which is also called methionine synthase. This enzyme requires methylcobalamin, a derivative of cobalamin or vitamin B12 for activity. Now, what you do want to watch out for here, again, is for the homozygous comps. They cannot tolerate methyl B12 because of their poor enzyme activity on the comp mutation. It's the ones that are homozygous that will have the biggest problems, and also their VDR status as well. But watch out for them. They don't require methyl B12 as much as the other B12s. If they normally have a B12 deficiency, you want to look at hydroxy B12 and adenosine B12 on the homozygous COMT individuals if you think that they are needing B12 with MTR. Also, sometimes they will require some, if you see homocysteine low and you've already ruled out oxalates as being an issue, they might require some very, very low dose SAMe. Now, the MTHFR gene, and I'll talk about the other B12 SNPs with this, even though they're coming a little later. MTHFR is methionine, and it's an essential amino acid required for protein synthesis. It is catalyzed by the enzyme methionine synthase. And methionine synthase eventually becomes inactive due to oxidation of cobalamin as a cofactor. Methionine synthase reductase regenerates a functional methionine synthase via reductive methylation. So depending on the MTHFR mutations, you also want to look at their B12 status. Now, the TCN genes, we have TCNs out there, and Ben Lynch is the one that had me look into this because he had a lot of people that, you know, they tested high, extremely high in B12, but when he went and tested their methylmalonic acid, they were low. It wasn't getting intracellularly, and this has to do with the TCN genes. These are transporters to the cell, and so B12 is really important to look at here. But if you see a high B12 and you see TCN, you probably want to go look at their homocysteine and their methylmalonic acid as well. And if those are elevated, they're still not getting it in the cell wall. So, you know, they say 1 out of 2 people, B12 status, you just don't know it's getting into the cell. So it's just best to do MMA, methylmalonic acid, and homocysteine on those individuals. Now, MMAB is, again, needed for cobalamin, B12, and people with MMAB can develop methylmalonic aciduria, and it is a disorder of B12 due to a defective synthesis of adenosine B12. So sometimes these people might require some B12. It's really good to look at MMAB SNPs. Also in this area, if you have anybody with MS, check their B12 status, look at MMAB. Also, you can look at MUT as well, because adenosine B12 is needed to help repair myelin sheath. And these people have a lot of central nervous system issues. So it's sometimes people with MS, they do have MMAB mutations as well as MUT mutations. With MUT, the deficiency in this enzyme is responsible for an inherited disorder of metabolism in the methylmalonyl-CoA mutase deficiency, which is one of the causes of methylmalonic acidemia. Again, many people with MS require adeno B12. It's good to look at their SNPs and maybe go check their methylmalonic acid levels. Common treatments for methods for MMA include liver transplant, liver and kidney transplants to combat the renal disease of methylmalonic acidemia. However, detrimental neurological effects can continue to plague patients even after a successful operation. Also, with many MS patients, when they're adeno B12 deficient, you'll see elevated cholesterol, and once you start giving them the adenosine cobalamin, their cholesterol can come down. MAT is methionine adenylyl transferase, and it's also needed for S-adenosine. It regulates the activity of S-adenosine methionine 2 by changing its properties to rendering the enzyme more susceptible to S-adenosylmethionine inhibition. It requires SAMe biosynthesis. All right, and then GNMT, this protein is encoded by this gene as an enzyme that catalyzes the conversion of S-adenosylmethionine to S-adenylyl homocysteine and sorkosine. Okay, AHCY is next, I believe. Just give me a second here. AHCY, I'm going to also give you a little information on what Amy Yasko talks about AHCY. It is the enzyme that converts S-adenylyl homocysteine to adenosine and homocysteine. Decreased AHCY activity should lead to lower levels of homocysteine. And BHMT, that one stands for betaine homocysteine S-methyltransferase. BHMT regulates homocysteine metabolism and converts betaine to homocysteine to dimethylglycine and methionine. Some people, they say, need betaine up in this area, but you need to also be careful with betaine in individuals who are homozygous COMT. I had to learn the hard way. I decided while I was in Hungary to juice some beets, since we don't have organic beets where I live here in America. I found some organic beets, and I said, oh goody, I'm going to go juice them. And I had 2 ounces of beet juice, and I was on the floor having panic attacks for hours. I didn't sleep for a couple of days, and not realizing that it can have an interaction, the betaine can have an interaction with people who are homozygous for the COMP158M and H62H mutations. But it can be very good for other people that don't have the COMT activity going on like that for the expressions. Now, DMGDH is dimethylglycine dehydrogenase, and it is a mitochondrial enzyme involved in the metabolism of choline, converting dimethylglycine to sorkosine. Again, we're hearing a lot about glycine, but I wanted to stop here and say a little bit about glycine. If you have somebody that does have oxalate issues, and they come back to you after you've given them glycine, where it can calm certain individuals, but if they come back with side effects, fever, edema, swelling, that could be a sign that there's some oxalate issues going on with those individuals. Again, B6 is really, really needed to be looked at before glycine is given to anybody, and then also their lysine as well. It's really important to know that glycine can help some individuals, but can really affect people that do have oxalate issues. Now, GAMT gene, this one's pretty interesting. It requires creatine. If you ever see somebody with that has the GAMT mutations, maybe 1 out of 20 will have heterozygous SNPs in GAMT. And then maybe 1 out of 50, 60 will have homozygous there. And you see that they have poor muscle tone. Creatine might be, you might want to look into creatine, and then also you might want to also check kidney function before you go give somebody creatine to make sure they have kidney function. Yeah, really has helped me. The creatine, knowing that I have GAMT mutation myself, has really helped me get a little muscle tone back that I've lost as I've gotten older. I do have the mutations. I did go get checked, and I was a little low on creatine. And I do have good kidney function, so I do take a little from time to time, and it's helped me out a lot. This is why they use it in bodybuilding, because it helps muscle. But if you do see somebody with poor muscle tone and you see GAMT, creatine is a good way to go with them. I guess back to Cynthia and Carolyn.
Thanks, Sterling. Okay, so It's really interesting. And so now if we're going to take a look at the methionine cycle, we've got our folate cycle. It's provided us with our 5-methyl, and now we've got to be able to use it and distribute it through the body. So if we look at the MTR, methionine synthase, it's vitally important in the utilization of our B12, because we have to remember that without sufficient B12, you're not going to be able to use your 5-methyl that you're creating. So we have a big problem when we see vegetarians and vegans. And in actual fact, what we do in clinic is every single person that comes in the door, we check their B12 levels. Now, serum B12 on its own isn't a great diagnostic marker. However, if you combine it with homocysteine levels and, as Sterling said, the MMA, then we get a better picture. But if you see really high unsupplemented B12 in the serum, then I would be thinking exactly what Sterling said. What's going on? Are there transcobalamin SNPs going on here? And quite possibly, yes. And then if we look at the MTR, also involved in the conversion back to methylcobalamin, but we need as cofactors SAM, our B2, and our B3. So these all work very synergistically, but probably one of my favourite topics is B12, because I can't believe diagnostically how much we are missing it. And if you're not checking B12, you need to be, because the amount of people that are on proton pump inhibitors that are reducing B12 levels is significant. And they're not on it for a couple of months. Some are on it for 10 years. And I recently had a very, very sick patient in who could barely walk. And we checked his B12 and they were so far below reference range. I mean, in Australia, we have our minimum reference range of about 150. Now, I can tell you if anyone's anywhere near that, they've got neurological issues. And this guy could not walk without a walking stick. So, and the doctors within the reference ranges were saying, well, the reference range is fine, but it's not fine. In Japan, they've just elevated the B12 reference range to be a minimum of 600. So we've really got to be looking at the B12. So then we assume then that we've got some good B12 and that we can create our methionine. And it's the methionine then that goes on to form, importantly, our SAM. And it's the SAM that is converted by the MAT gene, you can see there. And then SAM will donate its methyl to all our important methyl transferases, and we'll go into that in a minute. And in doing so, it converts to SAH, S-adenosyl homocysteine. But that is then broken down with the help of this AHCY gene, and then we get homocysteine back that is then remethylated with the help of B12, and so the cycle goes on. And you can see in the middle there, your choline pathway, that BHMT, is crucial in regenerating homocysteine back to methionine. And in so doing, importantly, creating our phospholipids for our cellular health, our cellular membranes. And as Cynthia said, the only elimination route for homocysteine in the body is through the CBS pathway. So if someone has elevated homocysteine, we have to be considering how much B6, but also be looking at our folate and our B12 in the process. So as Sterling's just gone through the key B12 SNPs, our MTR, MTRR, TCN, MAB, MMAB, and MUT, and the latter two affect our adenosyl cobalamin. So those of you who don't know, we've got our synthetic cyanocobalamin, which is just like our folic acid. It's synthetic. It doesn't exist naturally in the body. It's attached to a cyanide molecule, so it's actually not the best form to use. But it's our hydroxocobalamin that is the precursor to adenosyl or methylcobalamin. Adenosyl is our mitochondrial B12, whereas our methyl is our neurological B12. So we do need both. And once we have a look more at the CBS pathway, you'll see how integrally important the adenosine is feeding into that citric acid cycle. So MTR, as everybody said, it's methionine synthase. You'll see that written as either MTR or MS in some of the older literature. And really this mutation, if they have a mutation, it is a reduced function. And as you know, if we don't have this MTR working, then homocysteine is not going to be remethylated back to methionine. And so what happens is it builds up in the bloodstream. Our amount of methionine is going to be reduced. That then has a knock-on effect to SAMe. And we can often see homocysteine being excreted in the urine at high levels. I think the key gene that we need to be looking at here is the RS1805087, and that's also written as A2756G, and that's associated, as Cynthia said, with birth defects, particularly when it's associated with the MTHFR and Down syndrome. So here's that pathway in a nutshell, and you can see there that homocysteine is converted to methionine with the help of the MTR and obviously our B12, and our B12 really needs the MTRR. So those two really work in unison. So if we look at MTRR, methionine synthase reductase, as the name suggests, it really works with the MTR gene in ensuring that we have good methionine levels. And without that, we're not going to have good effects on our SAMe, so we're not going to have enough SAMe. The variant that we most look at here is the A66G or RS1801394. And again, particularly when it's associated with the MTHFR gene, is associated with the increased risk of neural tube defects and Down syndrome. And just another point on, I guess, the whole vegan, vegetarian, a lot of our vegans and vegetarians say, well, look, I get B12 from my food. Impossible. If you are a vegan or a vegetarian, there is no way that you will get sufficient B12 from your food. Impossible. It just does not happen. So, for some practitioners, that's a real battle. Some people, you're never going to talk them into eating any meat, but you've absolutely got to make sure that they've got either both forms of B12 and or they are supplementing with something. The MTRR, you can see there in the middle that the MTRR work in unison to help our B12, and you can see there how it all interrelates with the other genes. The MTHFD, vitally important for our folate cycle, and then we've got our CBS that runs off that. So as I said, the key mutations here are the 1805087 and the 1801394. So it is involved in some of the clinical trials with schizophrenia. Just on that, so is DMG-DH, very clinically related to schizophrenia. And I'll explain why a bit later when we tackle that gene. And I think the key thing there is obviously prostate cancers, because it does affect our DNA methylation, and the MTRR as well. They're both working in unison, so a lot of the studies you'll see will be these two working together and or in unison with the MTHFR gene. So the transcobalamins. So these, as Sterling said, they're transport proteins, and they take the cobalamin from the bloodstream to the cells throughout the body. And the cobalamin, as I've just said, is really only found in animal products. So meat, eggs, fish. It's not in vegetables. Does not exist in vegetables. So that's an important thing to remember. There's more than 20 mutations in the TCN gene, and it will cause a transcobalamin deficiency. And the problem is that if you're not getting these cobalamins from the bloodstream into the cell, then you're going to have difficulty gaining weight, particularly in babies. This failure to thrive can very much be associated with this. Vomiting, diarrhea, a shortage of really any of our red blood cells, and neurological problems. And some of the mutations can actually be almost a complete lack of transcobalamin. So a key sign is if you are seeing very high serum and you've got these TCN mutations, then you've really got to be considering that there probably is, there's not enough B12. So you're obviously not going to look at gut function or a pill. You're going to look at other forms of absorption of B12, and perhaps that's an oil or a cream that you put on the body. Because they're not going to get it if gut function. And that's another important thing to remember, that B12 is just so integrally important. Its absorption is so entwined with gut function. If gut function is not working, even without a TCN deficiency or any of these MTRs or MTHFR deficiencies, then you're going to have a problem with absorption. And of course, intrinsic factor is vitally important for that. And the TCN3, it relates to intrinsic factor deficiency. So here's the pathway again. I'm going to actually flick to the next slide because I think it just talks about this a little bit better. So the way this works is this, and it's important for us to understand the absorption of B12, both mitochondrially and through the serum, is that the cobalamins have to be released from the protein, right? So we have to be eating the animal protein. B, we have to have enough acid or pepsin in the stomach. That's why our gut function is so important. So dietary cobalamin then is bound to these proteins in saliva and our gastric juices, and you can see there it's represented in red by R cobalamin. And in the duodenum, that's the intrinsic factor. And in the blood, it's transcobalamin. So transcobalamin is then recognized by these transcobalamin receptors that are found on the cell surface, and you can see them there. And it's endocytosed into the lysosome. So it's only released if you've got good enough pancreatic enzymes. So it explains why the whole process of absorption might be damaged just by a pancreatic insufficiency. And we can see pancreatic sufficiency when we're doing our complete digestive stool analysis. And so once the absorbed vitamin B12 is transported into the various cells, it's bound to a family of glycoproteins, transcobalamin 1, 2, and 3. And any excess vitamin B12 is normally stored in the liver. But in the cytoplasm, it goes on to support our methionine pathway. So here, G is representing MTR or methionine synthase. E, you can see on the right-hand side there, is representing MTHFR. And in the cytoplasm, cobalamin B, so you can see this mitochondrion down the bottom. In the cytoplasm, cobalamin C converts dietary cobalamin to cobalamin, which is broken down to either the methyl side or the adenosyl side. So that's why it's such a tricky thing for people sometimes to absorb B12 efficiently. And so quite frankly, anyone with a digestive issue, I will give some sort of oil to support B12 because it's so crucially important for the methyls anyway. So B12 deficiency SNPs, I guess the key ones there is the 9606756, that's our transcobalamin 2. I don't know, Sterling, if we've got GIF on the variant reporters at the moment, but that's written as TCN3. and you can see that they're involved in, some of them, cancer, because again we need to have good levels of B12 to use our methylfolate. So in terms of our nutrition cofactors, obviously the TCN2 we need to be making our methylcobalamin, but our nutrient sources, our protein, it's eggs, it's beef, it's lamb, it's all those things that we really need to get our B12. And it's one of the things that I spend a lot of time with vegans and vegetarians, getting them to understand that they've really got to do something about it. So signs and symptoms, any and all of these can be present. And certainly ataxia or issues walking in a straight line is actually a really good sign. And I know from my mother-in-law, who all of a sudden started walking to the right, couldn't walk in a straight line. And when I asked the doctor to check her B12, it was seriously low. So I could talk about B12 all night, but it's certainly one of the things that we've got to be looking at. And in the test results, the lab ranges are just so wide-ranging. There's a great book by Sally Patchlock, and her book is called Could It Be B12? And she believes that there's enough evidence to suggest that really we should be looking at B12 levels closer to the 1,000 picograms per milliliter, or that's equivalent to 800 picomoles here in Australia. And I really tend to agree with her. Clinically, I'm looking for a minimum of a 600 picomole level of B12 when I'm considering B12 levels. And I think elderly, they're particularly vulnerable because their gut function is not as good and their diet definitely is not as good. So methionine. So methionine is so super important here. It's one of our sulfur amino acids. It's a key antioxidant, a free radical scavenger, but importantly, when I do testing for amino acids, I invariably see a very, very low methionine. So what's going on? Is there not enough B12? Is there not enough methylfolate? Is gut function in stopping the conversion of our B12 so you can create methionine? Are they a vegan? Are they a vegetarian? There's so many things to consider with methionine. And if it's low, then obviously SAMe is affected. And all of our methyltransferases that rely on SAMe then are going to be affected. So this is the one that I showed you last week, and I just think I wanted to talk about this because it is so important, because you can see that methionine is metabolized to homocysteine. And if the hepatocytes express betaine homocysteine through that BHMT, then it'll use betaine as a substrate for our methyls, and that's really important, and that's crucial. But when there's oxidative stress, we don't use the BHMT. We divert homocysteine down into the transsulfuration pathway, so you can see there with a red arrow, because we need to supply cysteine to keep glutathione levels high. Now, if that happens, we're inhibiting methionine synthase. So that's really important. We're inhibiting methionine synthase in oxidative stress. And so B6 then obviously becomes the important nutrient, because if we've got a functional B6 deficiency, there's going to be an accumulation of homocysteine anyway and a failure to maintain glutathione. So as Cynthia said, homocysteine, you'll see wildly ranging reference ranges, and it could be anything from 5 to 15. And I think from a functional pathology point of view, we're all in agreement that we should be seeing homocysteine sitting between, say, 7 and 7.5. That's absolutely perfect. Any higher, we know that the B6, the folate, B12 are going to be insufficient, and too low, we've got to be considering, okay, what is going on? Is there low methionine? Is there low SAMe? or is there major oxidative stress? So here we've got, so probably a good time here to talk about SAMe, because as we all know, it's our universal methyl donor for almost all biochemical reactions, including methylation of more than 80 different molecules, to support DNA, RNA, proteins, histones, neurotransmitters. And after that methyl is transferred, say, to DNA methylation, we then convert SAM to SOR, but that's an inhibitor of DNA methylation. So that's why we often look at SAM-SOR ratios, because SAM definitely depends on 5-methyl, betaine, B2, B6, B12, and a decrease of these is obviously going to be cause a decrease in SAM. But it's the conversion of methionine that we've got to look at, because we need those MAT genes, the methionine adenylyl transferases, to be working optimally. And so if we look at our MATs, then there's 3 key MATs that we've got, 1, 2, and 3. MAT1 and 3, they're predominantly found in the liver, and they are encoded by our MAT1A gene. And the effect will often be a high methionine. I very rarely see a high methionine, but I will see MAT genes 1 and 3 homozygous. But I know then that the SAMe is going to be important for those particular patients. MAT2, that's obviously encoded by the MAT2A and 2B genes, and it's found mostly in fetal liver and tissues of many organs, including our lymphocytes. And so 60%, though, we've got to remember, of all our methyl groups are derived from choline in the liver. And it's interesting that there's been a lot of experimental studies that show that if you've got high methionine and low MAT activity in the liver, but normal MAT activity in the red blood cells and the lymphoid cells, then you've probably got to look and see that MAT1 and MAT3 are defunct. They're not doing what they should do. But as I said before, clinically we're seeing more low methionine. And I think I used to be horrifically terrified of using SAMe in clinic, because I always thought that, you could have a really bad reaction. But I know now that obviously the 5-methyl can create just as bad, if not a worse. But what I have found in clinic is that if you're seeing the homozygous MAT SNPs, even with a COMT, I do tend to use very, very low dose. And when I say low dose, 100 milligrams of SAMe with quite good results, because a lot of the people with the COMT, they're not able to utilize their methylation, their methyl groups effectively, so they build up, and that's why they get the effect. But if you can support them with phosphatidylcholine, that'll help use up the methyls and give them just a fraction of SAMe, they do tend to do quite well. All right, so let's look at the MAT1A SNPs. So as we've said, these are really important because they're looking predominantly at our DNA, our proteins, lipids, and neurotransmitters. And that's that MAT pathway. So I think we all know that it's the methionine to the, by the way, for those of you that don't know, ADO-MET is SAMe. So we're looking at that conversion methionine to SAMe. And MAT2B, again, it's important for our conversion of methionine to SAMe. There's not a lot of research on the MAT2 in terms of the various SNPs, but as I said before, it's crucially important, more so probably from a pregnancy point of view, because it does have a big effect on our DNA methylation, and that's the pathway for MAT2. I think it's probably a good time here to talk about our methyl transferases, because these are, when you consider that SAMe supplies all our methyltransferases, and methyltransferases are simply enzyme groups that need this donation of a methyl to work. So anything that ends in an MT in the body is a methyltransferase, and it needs SAMe's help to donate that methyl group. So it doesn't really matter how we get these methyl groups, whether it's through SAMe or through 5-methyl or whatever it is, it's important that our body gets them. And if you consider COMT, so catechol methyltransferase, we're going to talk a lot about that. It really degrades our catecholamines, so it has a big influence on dopamine, epinephrine, and norepi. We've got PEMT, phosphatidylethynolamine, N-methyltransferase. So it converts our phosphatidylethanolamine to phosphatidylcholine. So it's going to have a huge impact on the ability of SAM, because remember we said 60% of our methyls are derived from choline in the liver. So we need that to be working. PNMT is phenylethanolamine. And so that's found in the adrenal and the medulla, and it converts norepi to epi. We've got GNMT, which is our glycine, so we need that to synthesize our N-methylglycine. GAMT, and the list goes on and on and on. HNMT, histamine methyltransferase, really crucial in breaking our histamine down intracellularly. So these methyltransferases have got to get their SAMe, and if they don't, then a lot of bodily functions are going to be compromised, and quite hugely. So I think we've had a chat about homocysteine. So we'll move on to GAMT now, and I wish I'd put that slide back in so you could see the GAMT. But you'll see it on your that pathway planner of Ben Lynch's, and just how critically important it is. Because it's, as Sterling said, it's really required for our creatine. And if we look at it, it stands for guanidinoacetate acetate methyltransferase. So like a lot of these, it's expressed in the liver. But if we're not creating our creatine, then we're not going to have correct energy. It's vitally important for providing energy for our muscle contraction. So it's really important for nervous system function. And it is really crucially involved in fatty acid oxidation. So I would be certainly looking for anyone that has issues with their cells, their energy cycle. I think you need to be looking at this GAMT. And as Sterling said, there's quite a few mutations. There's 49 mutations in this. And some of them, at the extreme, are involved in intellectual disability, seizures, and I think, we've got to be considering this in conjunction with some of these other SNPs. And that's just a diagrammatic example of, where this gene sits. And you can see there it's really involved in that whole SAM-SAW relationship. And, it's not 95% of our creatine is in our skeletal muscle. And so, we've got to be getting sufficient amounts. And just being able to see the amount of people that you see with low creatine levels on their blood reports, and particularly if you see it with nitric oxide synthase mutations, I'd really be investigating that a lot more, because as you know, that the urea cycle is integrally involved in creatine synthesis. PEMT. So this is our key gene that converts, the key enzyme that converts phosphatidylethanolamine to our phosphatidylcholine in the liver. And when you think that what does the PEMT gene help, it certainly helps us utilize choline. It's all these phosphatidyl lipids are really important for our membrane structure, bile secretion, our low-density lipoprotein secretions as well. So if you imagine that every red blood cell is surrounded by this nice fatty lipid layer made up of phosphatidylethanolamine, phosphatidylcholine, and phosphatidylserine, if we haven't got sufficient fatty membrane, then our absorption of our fat-soluble nutrients, A, D, E, and K, are going to be compromised. Brain function is not as good as it should be. And I think if we also look at, I think actually phosphatidylcholine is probably one of my favourite supplements, because I really think that a lot of people have significant issues with their cellular membrane health. And if we look at the pathway, we can see that our phosphatidylethanolamine is converted to phosphatidylcholine to produce choline, obviously to help bile synthesis. Very important too in fat metabolism. And you often see people who have had terrible time trying to lose weight that actually do start to lose good amounts of visceral fat and that difficult to move fat when you give them the good fat, which is the phosphatidylcholine. And obviously it's key for our membrane stability and our resistance to, viruses and bacteria, because we've got that great fatty bilayer. It certainly helps us add protection. Here's our PEMT SNPs. That's the key one that there's research on, but really it's there's probably not a heck of a lot of research on each of the individual SNPs. So the key things here we're talking about is obviously we've got to have good amounts of folate, good amounts of B12, B6, B2, betaine, glutathione, and make sure we're having sufficient methionine. The AHCY, this is key because it's definitely, that side chain that you look at, and if we look at this one, you'll be able to see better. It goes on to produce our adenosine and our inosine and recycle homocysteine back. And so if we see mutations, we're probably having reduced activity of SOR, S-adenosyl homocysteine, and so it's not being converted to homocysteine and adenosine sufficiently. And when SOR builds up, don't forget, it inhibits our methyltransferases. So important thing there to remember. And here's that pathway planner again, where you can see, excuse me, the importance of the AHCY gene in particularly also uric acid production. And inosine, if we're trying to create good amounts of adenosine, then really interesting, a lot of research has looked at it as an anti-inflammatory, particularly for neurons and glial cells, because it helps suppress inflammatory cytokines. So it is quite neuroprotective. It acts as a vasodilator. So I think we certainly need to be looking at this gene, and I know that's something that Ben Lynch is really looking into, the adenosine. Unfortunately, we can't really check for that, but you have some great tests in the US that you can check for adenosine levels. BHMT, we've talked about, this is what we call the shortcut route through to converting homocysteine back to methionine. And really SNPs in this pathway are associated with just a decreased conversion of homocysteine back to methionine. And the key one in this group that I tend to look at is the BHMT08. And I can't find any clinical evidence other than we've got betaine in the way of beets, but we've also got betaine hydrochloride that we use for gut function. And if I see a BHMT08, I usually support it with hydrochloric acid and digestive enzymes, and we actually even see a really good change in mood. And there's the key SNPs there that you can see. As I said, BHMT, it definitely has a huge effect on mood, and if you can support it with the betaine hydrochloride, it actually does have huge improvements. Choline. So choline, as we've said, crucially important for our methylation, almost on every level. And it's really needed for structural integrity, our cell membranes, etc., etc. Now, the DMGDH, it's only a gene that I've only recently started looking at, but it does help with choline catabolism, and you can see on the pathway planner, it's the end enzyme on that choline SNP, and it forms sarcosine. And deficiency, I guess key deficiency symptoms you might see, someone might say, I have a really fishy odor, or I've got muscle fatigue, or increased serum creatinine kinase would be the key things that we would look at. And I have done quite a bit of research on this, looking at the association between this and schizophrenia. And I've started to use some sarcosine with some patients who have that real excitability, because it tends to create an excitability of nerves. And initially, people do say that they have some really good results, and they actually, for the first time in their life, they feel calm. So this is probably an evolving one that we will spend more time on later on to look at. But initially, I'm seeing some really good results from using sarcosine with these people. And there's the key SNPs, and you can see that there's association with fertility issues. And GNMT, Sterling's already spoken about this, the glycine N-methyltransferase, and it really helps control the relative amounts of SAM and SOR. It's almost like a backup for your histamine, your methyl levels. So it provides a backup source. So if people have got these mutations, then you might see that when they do get stressed, and there's a reliance on methyls to get them through that stress, they don't have enough backup to help them get out of the stress response. Okay, I think we did really well. It's exactly 1 o'clock, so we'll open up to questions now if anyone has any.
So great presentation. Found out some interesting things for myself as well there. Thank you. So now that we've got to the end of the presentation, we have actually got some questions that have come through. So I'm just going to get those questions for you. So bear with me a second. Here we go. So let's see. Okay, Thomas is asking, this is sort of going back to nearer at the beginning. So Thomas says, I understand that over-methylation is bad. Can someone explain how, if methyl groups are so great for us, we can get too many? How and where do they jam up the system?
Okay, I'll start, and then Cynthia, you can add. I think, so what happens, sorry?
I'd like to add COMT at the end, give you a prime example.
Great, all right. So basically over-methylation really means that the person is unable to utilize methyl groups. So that when you are putting, let's use 5-methyl tetrahydrofolate as an example, if they can't utilize it and you're giving them 5-methyl, because naturally they're going to create methyl groups anyway, because if they're eating leafy greens, that's going through the system. So they're creating their own, but their biochemistry is such that they're not able to utilize them. And so when they can't utilize them, they build up. Now, the utilization may be, there's not enough choline, because you need 3 methyl groups to create a phosphatidylcholine. They may not have enough B12 to transport it, or they may have the COMT gene, and Sterling will tell you a bit more about that. So the problem is, when we say over-methylation, what we mean is they can't use the methyl groups they are creating. So if you can help the utilization of them, you will then solve the problem. So that's what it's about. It's about the utilization of them.
I just want to add one comment. I've had people come to me who have been on a pharmaceutical drug called Deplin, Deplin, I think, is 7 or 15 milligrams of 5-methyl tetrahydrofolate, and they've been able to tolerate it. Once we start opening up the transsulfuration pathway, the methionine homocysteine pathway, their need for that Deplin lowers dramatically. So in my personal experience, when I'm working with people, we open up all the pathways first, and then we'll start to layer in folate, because most people don't need as much as they think they do. Some people do, but a lot of people who think they need a lot don't. And so to me, a lot is 800 micrograms a day.
Yeah, I agree totally.
Sterling,
Okay,
do you have anything?
the COMT, I'd like to add, you know, when you're taking any of these methyl donors, they will increase epi and norepi. And if you see somebody homozygous for V158M and H62H, they 40% of that is impacted for them. They're not going to be able to break it down. You can actually put them in mass panic attacks. I have been doing this for years now, and I've become a support hotline for many that have gone to doctors and have gotten high-dose methyl B12 along with methylfolate, and call me at 2, 3 in the morning out of a dead sleep. And I am their only support system to keep them from jumping off the edge of a building or committing suicide. So you really need to take things like that into consideration. Also, you want to look, again, I'm going to repeat this, oxalates, they're really big. If you ever see low methionine, 90% of all people who are chronically ill with low methionine, low homocysteine, have oxalate issues. You look into their B6, you look into their lysine, okay? You give those people jammed and their homocysteine's already low, high-dose methyl B12, methylfolate, you're going to really lower it. And you think that they're wigged out now, they're really going to be crazy. They're going to be out of their mind. They're not going to be able to think straight. So you've got to open up different things first and take other things into consideration before ever doing that. I am an example. I ended up in the hospital 6 days on morphine. It opened me up so fast. So everybody's biochemistry is going to be different.
Yep, I agree. I would support that 100%. And I think also, too, we've got to, if you're considering giving someone methyls, as Cynthia said, you've got to make sure that the other pathways are working efficiently first. And it's often the last thing we give, and some people we don't give it at all. So I think that's important to remember. Just because they have the MTHFR gene, it does not mean they need any methylfolate. So I think, and looking at the COMT gives you a really good indication as to, and the MAT genes, as to whether they really need methylfolate at all.
Okay, thank you for that. We have another question here from Tara. Is MTR and MTRR an upregulation or downregulation of the enzyme, increased or decreased activity?
Down.
Okay, that was short and sweet.
Yeah, they're down regulations of the enzyme.
Okay, excellent. And then just as a further on from that, does an MAT SNP cause decreased activity, less conversion of SAM to SAH?
Yes, it does. So generally they're a down regulation, and in some of the SNPs they are a complete block of the conversion of methionine to SAMe.
Okay, so that is all the questions that we've got at the moment. I'll just give you a second or two to type in any other questions if you've got them.
I'll just, while you're doing that, I'll just, we might all just add something. But the thing I wanted to say is, if you don't know the signs and symptoms of people who are not coping with methyls, the key things to look at are muscle aches and pains. And probably, I would say 95% of all people who start with methylfolate will get that for the first few days. And we would expect that that would decrease. Headaches, the same thing, and that's very common. There's rashes. So if someone suddenly gets a rash all over their body, you've got to be thinking perhaps it's methylfolate. I've had some people that have even had seizures, And as Sterling said, very high anxiety in a lot of cases, and often severe depression, suicidal. And we've had people in the clinic. And so you all know that the antidote for that is to give some straight niacin. So we would normally start with 100 milligram niacin and give it to them every half hour if we have to. They will flush, but it doesn't matter. And if you can give them a food or meal, it'll stop the flushing. But the niacin, if you're a bit concerned about someone, always give the niacin with the methyl, so that you explain to them the signs and symptoms, and you give them the niacin.
Okay, so whilst you were doing that, Caroline, we've got a few more questions come through. So we've got one here from Donna. Could you say more about Sarazine? It was mentioned at the very end. Is that a supplement?
Sarcosine?
Sarcosine, yes.
Sarcosine. Yes, sarcosine is just an amino acid. And the clinical trials in schizophrenia, they use a dose of 2 milligrams BD twice a day. But I've really been just using, say, 500 milligrams twice a day, or sometimes 1,000 milligrams twice a day. So we just get it compounded from the compounding pharmacy. But I don't know if you can buy sarcosine as a supplement over the counter in the US. I'm not sure.
Okay, and just to carry on about the niacin, how does the niacin lower methyl groups?
Cynthia,
Well, it helps break down dopamine, epi, and norepi, and that's some of the rise you'll get in people who have COMT issues and trouble breaking down those enzymes. COMT is niacin dependent, so people who have homozygous COMT tend to use a lot of their niacin up. So you're really going to use all their niacin up when you give them high-dose methyl donors and they have that kind of reaction. So you want to get it back in. I've actually, that's when people are given epinephrine with COMT and go into anaphylactic shock, they should always get IV niacin after the anaphylactic reaction's gone. It breaks down dopamine, epi, and norepi and calms them down.
Okay, and Donna's saying here, instead of niacin, you can also purchase NADH, Mojo by Anada, as a supplement here in the US. That seems to be great to bring down over-methylation. Any comments on that?
Cynthia?
I didn't hear what that supplement was.
It's NADH.
Oh, NADH?
Yes.
NADH can actually cause an increase in dopamine in some people. So again, homozygous COMT, you want to try to be careful because NADH can cause an elevation. I'm homozygous COMT and I do have it expressing for many reasons, but anytime, and you can go look in literature, anytime you do give NADH to calm down somebody that may be over-methylating, look at that COMT again, because in their neurotransmitters, if their dopamine's high, you don't want to raise it anymore. I can do a sublingual NADH, very low dose, and have panic attacks for a week because my dopamine will go up that high.
Yeah, I must admit I've never used NADH for that purpose. I only ever...
Then you've got to look at the NADH and then the NADPH ratios too. So, chronic fatigue people usually have NADPH deficiencies.
And I guess it would also depend on the NDUF genes as well, Sterling.
Yeah, so I mean, if they were fine on the COMT and you saw that, and you knew that dopamine, epi, norepi was okay for the most part, it might be okay. But for somebody who has a family history of, schizophrenia and stuff in their family, may not be the best one for them.
And I have to say, I've just mentioned one thing. NAD, not NADH. NAD IV therapy is often used to treat alcoholism and drug addiction. So there's an interplay with neurotransmitters there as well.
Yes.
Okay, great. Well, that's all the questions that we've got. If you do have any further questions, then please feel free to email them through. We're just getting some comments to say thank you very much. This has been a great webinar as usual. So, and I echo that. It has really been it's been a very informative webinar today. Just to let you know that next week we're on webinar 5, which is the transsulfuration pathway. The dates for that again are the same dates the same times next week. Tuesday the 27th for the US at 7:30 p.m. and Wednesday the 28th of October at 11:30 a.m. So I want to say thank you for all of those that attended today, and thank you very much to Sterling, Cynthia and Carolyn for providing some great knowledge once more. So thank you very much everybody, and